Kind of feeling the pressure of going first here, so hopefully I can keep you guys awake and entertained for the next 20 minutes. This has managed to keep me occupied for the last year, maybe a little bit more than that now. So yeah, hopefully 20 minutes you guys can find something interesting in here. So a little bit of background on my research here. Aquatic insects actually provide a lot of information about streams. It's kind of common sense. Obviously, the abundance is going to serve as a relative indicator of nutrient richness. If you don't have a lot of insects, there's probably not a lot there for them to eat. More insects, there's probably a lot of nutrients there. But it might be less intuitive to realize that the presence or absence of certain insect species can actually tell you whether there's pollution present within a system or not. So pollution tolerance varies greatly between species, and one common technique that ecologists use is the biotic index. And it basically ranks a bunch of different insect taxa on a scale of 0 to 10 according to their organic pollution tolerance. You can see an example I threw up here. Odonata is the order of dragonflies and damselflies. There's a nice little blue damselfly that I managed to snap a picture of. Enelagma, and you can actually see they have quite a high pollution tolerance, 7.9 out of 10. So one drawback of this technique is that it requires insects to be identified as specifically as possible to the lowest possible taxon. Morphological identification has a bunch of disadvantages, and I found this out the hard way. It requires a lot of training. It's extremely time-consuming depending on what kind of insects you're trying to classify. There's a lot of unclear features. I had a great key that I was going through, but still, when you have tiny little insects and you're trying to figure out, you know, how many hairs are on their legs or something like this, it's just really difficult and time-consuming. So I'm particularly interested in Plecopterans because they have really low pollution tolerances because of their high oxygen requirements. You can see here pollution tolerance values for a few of the genera, and compare that to, say, the dragonflies. They're quite a bit lower. I don't think there's a single one that's higher than 4 on there. So a little bit about Plecoptera. They're a primitive order of insects commonly known as stoneflies. They spend most of their lives as aquatic nymphs before they emerge as adults, and I would actually be surprised if anybody here who's not an angler or an entomologist has ever heard of them. I actually hadn't heard of them before I started this research. And I find them really interesting because they're extremely diverse. They have a huge range of sizes, behaviors, life cycles. I have some pictures on the next slide here. You can see this one belongs to the family Plecopteran. It's about 7 millimeters long. This guy here, I don't know if you can make out that ruler or not. That's Pteronarxus. He's about 4 centimeters long. So there's a huge range of shapes, sizes. I threw this adult down here. Has anybody ever seen one of those flying around before? No? They don't live for very long. They kind of emerge just to mate, and then they die. Start the life cycle over again. So yeah, family Plecopteran I was really interested in. They're cold adapted, so obviously they're dominant in this region. And this actually makes it really hard for us to try and key them out because, like I mentioned, they're these tiny little guys. They lack distinguishing characteristics, and when you're going through a key, there's 29 different species to choose from. So enter DNA barcoding. It's been gaining popularity as a fairly accurate method of species identification when compared to a good database. It takes the 650 base pair region of the cytochrome C oxidase gene, and basically you sequence it, throw it into a good database. We use BOLD, or Barcode of Life database, and it usually gives you a species identification. This was a paper I stumbled across when I started this research from 2011. Great paper, had a lot of really important conclusions, found that DNA barcodes do increase the number of taxa. As you can see here, It increased the taxonomic inventory by 475% over an amateur sitting there and classifying these things out. So compared to me identifying these by morphology, they'd find almost 5 times as many different species. One thing I didn't like is that you'll notice here, under Plecoptera, they only had 47 specimens, 2% were identified to the species level. 98% higher than genus. And as you can see from those pollution tolerance values, something higher than genus doesn't really give you a lot of information. So my goals with this research were to confirm the validity of DNA barcoding as it applies to Plecopterans, to, after confirming that, to use barcoding to identify any ones that I couldn't identify by morphology, and also to compile a checklist of species in the area to serve as a baseline for the future. I don't want to stir up any controversy, but I'm sure everybody knows that we're probably getting a pipeline through here sometime soon. And it's a good idea to have a baseline so that we can monitor any changes. I can tell you 100% for sure, if stoneflies start disappearing from that river, there's probably some pollution going on there. So for my methods, we collected a total of 357 stonefly nymphs from these sites on the Crooked River just north of Prince George. It's about an hour drive. You can see Summit Lake down there, Bear Lake up there. Has anybody ever been for a drive north up there? You know where this area is? Yeah. It's nice up there. Anyways, yeah, we sampled periodically through the summer when we could after the ice melted and after the floods kind of subsided for the year. Just sampled them using kick nets. So you stick a net down in the water, kick up the rocks, and everything kind of floats downstream into your net. Sorted through, picked out all the stoneflies, put everything else back. Preserved the stoneflies in 80% ethanol, and then I examined them back in the lab under a dissecting scope and tried to key them as specifically as I could. There were 4 species we got that I was able to identify with a really high degree of certainty based on various distinguishing features. And we decided that we're going to use these as quality controls, run them through bowl, see what we get back. If it matches, then we can say that, you know, this technique's working fairly good within the Plecoptera. And like I said before, once the validity of barcoding was confirmed, we applied it to ones that we couldn't identify morphologically. As for the DNA extractions, this technique was actually suggested to us by Sarah Gray, so thank you very much. Basically, just cut off a piece of the leg of one of the stoneflies, chopped it up into thin sections, put it into this Helix solution, boil it, and you get DNA out of it. And then you throw that into your PCR reaction. PCR. The R stands for reaction. That's kind of redundant. So, for the PCR, we use the untailed Fulmer primers, LCO 1490 and HCO 2198. These are the ones that Fulmer originally used in his paper in 1994. I don't know if you can see this gel very good, but we confirmed the presence of the product, cleaned the samples, and submitted them for sequencing to UNBC Genetic Facility. For the results, the 4 species that I was able to identify with a high degree of certainty were Clocenia sabulosa, Pteronarxis californica, Hesperoprella pacifica, and Zapata cinctipes. 3 of them, Californica, Pacifica, cinctipes, gave me a 100% match in bold. I was thinking, yeah, this is great, this technique's working. Threw in Clocenia sabulosa, no match. I wasn't sure what was going on. It's a really common species throughout all of North America. Took another look. Turns out there's no record for Clocenia sabulosa in the database. So I took some pictures of the specimen, uploaded my sequence to Bolt, and it's since been accepted as a barcode compliant record. So if you go and throw the sequence for Clocenia sabulosa in there, you'll get this screen. You can see single match in Clocenia sabulosa, and if you click this little publish guy, you get to see my picture there and deposit it in University of Northern British Columbia. So as far as I know, this is the first record that UNBC has on Bolt, so I was kind of excited about that. All right, on to the rest of the results. We selected 35 specimens for sequencing and analysis. There were a bunch we decided not to do for various reasons. Like the Hesperoperla pacifica, we had over 100 of them, so there wasn't a point sequencing every single one. So we kind of took these as a representation of the larger sample. So you can see Placenia originally had no match. Everything else, 100% match all across the board for our 3 test specimens there, and then we identified some new ones. And then this came up. Squala, 98.25% match. So we have 5 specimens that I had keyed out as belonging to the genus Squala, had a 98.25% match to Squala americana. So Bold's confirming that I'm in the right genus, I think. But according to Bold, this isn't a high enough sequence similarity to confirm a species level match. To make things even more confusing, there's only 2 known species of Squala in North America, S. americana and this other one called S. curvata. Bold only has data available for S. americana. And another thing that makes it even more confusing is that these 2 species cannot be told apart as nymphs. They have to be sexually mature, and it has to do with their reproductive organs. So I did notice this is the tree that Bold gives you when you throw a specimen in. And as you can see here, our unknown specimen is definitely grouping separately from these other Squala americana. So Bold lets you see if there's private specimens in the collection, and they actually let me contact the owner of a private collection there. And I asked him if I could get Squala curvata data that he wasn't sharing at the time. And he was nice enough to send it to me, and I used it to build a phylogenetic tree. So you can see samples 14 through 18 here are unknown Squala. This is the S. curvata sequences that I got. So you can see it's definitely grouping separately from there. So I'm fairly certain we don't have Squala curvata. But you can see there's still a little split between these other S. americana and our samples. So what does that mean? Specimens are very clearly not S. curvata. They still appear to be distinct from S. americana. And before I get excited and call it a new species, there's a number of different reasons that this could happen. It could be geographic variation. I'm not 100% sure that that could be the case because, as we saw with all the other specimens, they were coming back as 100% matches to stoneflies as far away as California. So maybe that one, maybe not. It could be an unresolved species complex in which we have a bunch of bugs that look similar and it still has yet to be sorted out the genetics of the different species. Or it could be something amazing and we could have a new species or subspecies. But unfortunately, with only 5 specimens and the limited sequences that were provided to us, we really can't tell for sure at this point. So it's kind of anticlimactic, I'm sorry. As for the species occurrence, we were able to identify 9 species in 7 different genera. I constructed a kind of approximate timeline of when they're in the river. You can see Squala was only there for a bit. This Hesperoperla pacifica was interesting because we had it there almost every site, almost every sampling date. One that really interested me though was this Zapata that we found on June 7th. I don't know if you guys are friends with any anglers, but they have really imaginative names for bugs. So this Zapata, they call it the tiny winter black. Now if we had all day, I'd let you guys try and guess why they call it that, but I'll just tell you because I'm getting a card. They're tiny, they emerge in the winter, and they're black. So anyways, I found a paper from southern BC that found that they tend to emerge between January 8th and April 7th. And here we are finding them 2 months later, June 7th. So one interesting thing is that CR5 contains this underground spring, which is always feeding cold water into the river year-round. We were lucky enough to have an oxygen monitor with us that day too, and we noticed that that site only had 3.5 to 4 milligrams per liter, compared to the nearby site CR1, which had 10.7. So I'm thinking maybe the cold temperatures from the spring or the low oxygen content could be delaying their development. All right, so in conclusion, for the control species, barcoding resulted in IDs that were consistent with the morphological ones, suggesting that it is an effective technique within this insect order. The specimens that could not be determined by morphology, we were able to successfully identify using barcoding, further highlighting how useful this technique could be, especially in large analyses. So we had a new record submitted to BOLD, which I'm super excited about, because now that's going to be there for the rest of the time. Well, unless they disprove that barcoding works and take the site down or something. And we also were able to identify this kind of genetically distinct population of swallow, which I think, even though we couldn't come to a conclusion about it, it still highlights the technique's sensitivity for differentiating different species. So I want to thank Dr. John Sandberg, who gave me the sequence data from S. curvata and S. americana. So it was really helpful for trying to sort out that tree there. Of course, I want to thank Dr. Poirier for serving as my second reader, and along with Dr. Huber and Dr. Lindgren, met with us a couple of times to discuss entomology. And of course, I want to thank Donnie, who made this research possible by taking me out to get samples and, yeah, providing lab space and funding and all that fun stuff. So, thank you all for showing up.